To learn some PDB vocabulary, let's look at the PDB entry 2HBS. It's a hemoglobin structure, deoxyhemoglobin, but it is sickle cell, meaning it has that mutation. And so if you look at the deposited coordinates, the asymmetric unit, you actually see how that valine, which is mutated, sticks to another hemoglobin molecule. And this is the basis for formation of the fibers that then distort the red blood cells. What I wanted you to think about is that in this PDB entry, there are two hemoglobin molecules interacting with each other. Each of these hemoglobin molecules is considered an assembly. Within each assembly, there are 4 chains of 2 types. So there are 2 entities. There is only one small molecule ligand, the heme, and then there are waters. This is what is summarized in this figure, where the entry is the whole thing. The biological assembly is each of these hemoglobin molecules. There are polymer entities that are shown in orange and blue. Then there are non-polymer entities, the heme groups and the waters. And then there is instance. So if you are looking at this particular chain, you will call it the instance of that entity.